I have the honor of introducing one of your hosts for tonight's special lecture, Dr. John Mandelman, Director and Senior Scientist here at the Aquarium. Dr. Mandelman innocently took an internship, like many of us, here at the Aquarium in 2001 and has been here ever since. In addition to his responsibilities as the director of the research program, John is also responsible for an independent research program studying the effects of human-induced disturbances on the physiology, ecology, conservation, and management of various marine fish species, including sharks, stingrays, skates, both regional and remote tropical marine environments. Please welcome my colleague, Dr. John Mandelman.
Welcome, everyone. I'm sure you're all here tonight to hear my lecture on cod, right? You're 3 weeks early. That's on the 28th, but hopefully some of you will come back. I don't think we're going to have as big a crowd, though, maybe. Something just, I don't know. Something just tells me. But we're all here tonight to learn about some fascinating animals. And I wanted to start out just really quickly before we get to our lecture by just talking about a few things related to the aquarium's mission. Research and conservation are integral parts of our mission. For the last several years, we've been conducting research on sharks, as well as their close relatives, the stingrays and skates. And in addition to that research, we've engaged in a lot of outreach and even more recently, advocacy to help protect these animals. The research side is incredibly important, but before we can, for the reason that before we can protect these animals, we have to understand them. We have to know more about where they move, how long they live, how fast they grow, how they respond to stress, to human-induced stressors. We have to know about them. We have to go out there and work with them in the field before we can invoke protection plans and try to help these very vulnerable animals. So, because of all this work we've been engaged in, we've become very close with several colleagues in the region who do amazing work with sharks. And tonight we're privileged and fortunate to have some of the folks in the region that do shark research here with us to talk a little bit about their work and how they got to this point. Before we get to these lectures, I just wanted to introduce my co-conspirator for the evening. Cynthia Wigren is the founder or co-founder and president of the Atlantic White Shark Conservancy, which some of you may have heard of before. I'm doing incredible work that she'll talk to you about tonight a little bit before we get into our lectures. A couple of other notes before Cynthia talks. Our lecturers tonight are going to be, we have a full slate, and they have very short time slots, so please hold your questions till the end. We're not going to be introducing these 8 speakers with their bios or their titles. That's all on the sheets that you guys should have received when you came in. So we'll try to keep it as quick a transition as we possibly can. So without further ado, I'll have Cynthia come on up here.
Good evening, everyone. I wanted to start by thanking Gina and John, who I've been working with for the last year to put this evening together. Thank all of you for coming. It's amazing to see so many people here this evening. And to thank all of our scientists who are here tonight. We're really excited to learn more about their research. So a good segue into my organization is just in everything you hear tonight from these scientists, you won't really hear how much they have a challenge with funding the research, and that's a very significant part of it, is to find funding. I started an organization called the Atlantic White Shark Conservancy to fund white shark research on the Cape. I work closely with Dr. Greg Skomal and John Chisholm, who you'll meet tonight, to support their white shark projects. And this season, we're really excited about funding a first-of-its-kind white shark population study in the northwest Atlantic. In addition to funding research, we also promote shark education through community outreach and school talks. And one of our biggest education initiative that we launched this year is the Gills Club. You can see the logo up top and a bunch of little girls who came to an event that we did where they learned how to tag a shark and learned all about the science behind it. And that's them successfully releasing their shark into the water. Very excited about it all going very well as planned. So a big part of that organization is to connect girls with an interest in sharks to the science behind shark research. And we have over 30 female shark scientists from around the world who are participating in this program that we started. And our hope is to jumpstart their interest in science by giving them really strong role models in the field. We have 2 female scientists here this evening, and one of them, Heather Marshall, is a co-founder of the Gills Club. The other thing that you'll see on my slide is the license plate. So if you're a Massachusetts resident, a really great way that you can help to support the research is to pre-order a great white shark license plate. And you can do that on our website by downloading an application, and that's atlanticwhiteshark.org. So it's time to meet our scientists, and the first person that is going to kick things off is a good friend, John Chisholm.
Thank you. Wow, what a crowd. Especially on a night that the Bruins are playing the Canadians in a playoff game. But I'm guessing that there's a lot of people that like sharks out there, which is good because I really like sharks. So what these other guys. I've been a shark nut for as long as I can remember. By the time I was going to kindergarten, I had already made up my mind that I was going to be a shark biologist when I grew up. And I actually grew up only less than 5 miles from here. And fortunately, I was able to come to the aquarium pretty much once a month and stare at the shark wall. You can see this is the new shark wall. I'm glad that the aquarium brought it back. But I would come here and I'd see the nurse shark and the sand tiger sharks. And every once in a while, the sandbar sharks they'd have in the tank. But it was always the shark wall that fascinated me, and I'd stare at it and look at all the species and memorize them and think, how many of these species am I going to get to see when I grow up? How many of these species am I going to work on? And I always dreamed, you know, I'd be in Australia, or even better, on a deserted island surrounded by sharks. The last place I ever expected to end up was right here in my own backyard in Massachusetts. But fortunately, as I grew up, I learned that, you know, we have a quite diverse shark population here in Massachusetts. And it's surprising still when I meet people, they don't realize the diversity of the sharks we have. And here's just some photos of the different species you can find. One of the reasons we do have this diversity is location, location, location. New England is conveniently located right between the northern cold Labrador Current and the southern warm Gulf Stream, and it makes our waters really productive, makes for a nice nutrient-rich ecosystem. And in addition to that, it also has a wide range of habitats, from coastal estuaries to deep water canyons, benthic, pelagic. There's just plenty of habitat and just as many species to utilize them. In Massachusetts, out of the 8 orders of sharks, I don't have time to get into them, but out of the 8 orders of sharks, we have 5 of those orders represented in New England waters. And about 15 families, which is over 24 species of sharks. Unfortunately, I don't have time to talk about all of them tonight, so I'm going to real quick introduce a few, and you'll hear details on some of them as the night goes on. So the first group I'm going to talk about are the sharks that get lumped into the category sand sharks. The first of these is the spiny dogfish. It's the most abundant shark in our waters, probably the most abundant shark in the ocean, if you ask fishermen. It's a small shark. Distinguishing characteristics are 2 spines on each dorsal fin. Another small shark called sand sharks is the smooth dogfish. Now, smooth dogfish mainly stay south of Cape Cod. They like warmer water. They're about the same size as spiny dogfish, but they don't have the dorsal spines. And they also don't have very distinct teeth. They have pavement-like teeth, which distinguishes them from the third shark in the sand shark group, the sand tiger shark, which has the well-pronounced teeth. If you've been to an aquarium, you've probably seen sand tiger sharks. They're exhibited all over the world. A lot of you are probably saying, well, how's this? I know sand tiger sharks, they get pretty big. How are they lumped into the sand shark category? Well, as you'll hear in a little while, the sand tiger shark is making a comeback in Massachusetts and New England coastal waters and different estuaries, but we're only seeing juveniles. They're the same size as the smooth dogfish, the spiny dogfish. Fishermen who aren't used to seeing them lump them into the category of sand shark. it's important to know the difference, and here's a comparison of all three: spiny dogfish on top with the spines, smooth dogfish in the middle with no spines, no teeth, sand tiger on the bottom with all the teeth. And the reason it's important to know the difference between these sharks is the sand tiger shark is a prohibited species, the other two aren't. And unfortunately, fishermen associate the name sand shark with the spiny dogfish, which is considered a nuisance and often killed. So we've unfortunately been finding these small sand tiger sharks killed by fishermen that consider them sand sharks. So now you all know you can take that information and go educate your friends and family members that fish. It's mainly striped bass and blue fishermen that come across these sharks. Moving on to the larger coastal and pelagic sharks, we have the sandbar shark. It's another southern shark, mainly south of Cape Cod. It is often confused with bull sharks, so every once in a while you'll hear a report that a bull shark was caught from the beach on the Cape. It's undoubtedly this shark. We do have a couple of recent records in Cape Cod Bay, and one of the ways I look at the shark is it has an oversized dorsal fin. It's tough if you don't see a lot of sharks, but if you see them in comparison, the big dorsal fin really stands out. Oh boy, this is a blue shark. Probably the most abundant big shark in Massachusetts. This is a porbeagle shark. You'll be hearing about all these in detail later, so I have the real easy job of cranking through this. The porbeagle shark is in the same family as the white shark and the mako shark. You can tell it apart by the white patch at the base of the dorsal fin. It also has a secondary keel on its tail. Mako shark, one of the favorites of recreational shark fishermen, very streamlined, fast shark, can leap out of the water, likes to eat bluefish. Large makos will eat tuna and even marine mammals. Moving on to some of the oddballs that we have in New England waters, the Thresher shark, which is easy to tell from its long tail, which can be the same length of its body. It can actually use this tail with precision to basically stun its prey. And hammerheads, which we see scalloped hammerheads out in the canyons, out in the warm water. They're a shark that likes to school, and you can see them in this a school of them in this photo. Everybody can tell a hammerhead because of the nice flat hammerhead with all the electrosensory organs for detecting prey. Moving on to the big three. White shark doesn't need an introduction. I'm sure everybody here has seen the white shark. Of course, the resurgence of the gray seal population is making them the talk of the town here recently. Basking sharks, second largest fish in the world. Often confused with the white shark because they share the similar proportions, but this is a filter feeding shark. It's only really dangerous to plankton. And then we get to the whale shark, the biggest fish in the ocean. A lot of people are surprised that, we do see whale sharks up here in the canyons in the summer months, and another filter feeding fish. So why I do what I do? I love sharks, and I've had the good fortune of being able to work on a lot of great research projects and meet a lot of great people, some in the audience, some that you're going to hear from in a moment. And I've been with the Division of Marine Fisheries. I've been with the Division of Marine Fisheries for 23 years now. And although I feel like I've been studying my sharks my entire life, I actually didn't become part of the shark research program until 10 years ago this year, when I had the good fortune of working with one very charismatic individual, and I'm talking about Gretel, the white shark, at Noshon Island. Of course, I'm just kidding. You'll hear from Greg and the rest of the crew, and they'll fill you in on all the sharks that I just breezed through. And I just want to say, thanks for coming tonight, thanks for listening, and enjoy the rest of the show.
It's my honor and privilege to introduce my friend and colleague, Dr. James Sulikowski, as our second speaker.
All right, let's see here. So, why did I get started as a shark researcher? Well, from a very young age, I loved the ocean. and everything marine about it. And that is me with blonde hair. And with the bowl cut holding 2 catfish that I caught as an avid fisherman back in Ohio. And I've, ever since then, just been fascinated with those marine creatures. But to be honest with you, and I'm saying this for the first time here, I loved hippos growing up. And I really want to be a hippo researcher. That was until I found out that they were way more dangerous to study than sharks. All right, so my transition to the dark side was, fairly easy. And I've had the privilege thus far to study several varieties of different shark species. And my research has morphed, which has been exciting. I guess you would kind of coin me as a physiological ecologist. I'm really interested in linking life history characteristics with technology to better understand where they go and why they go so we can conserve and manage them in a more appropriate way. And so one of the things I'll talk about is some of our technology. And this is Amy Carlson. I'm sorry, Amy, she's in the back. She can wave. She's my geospatial technician and the right-hand person in my lab. And she's holding a satellite tag. And that is the smallest satellite tag that they make, and that's why she's so happy. Because these tags are wonderful. They record depth, they record temperature, and record movement patterns, horizontal movement patterns, so that we can get an idea of where these sharks go at certain times of the year. And she's so excited because this is a tag that we can put on baby sharks. And I'm going to show you a little video of some of our current research. And this is actually,
Click on the video.
Oh, okay. A baby porbeagle. And according to my colleagues, Lisa Natanson, she says it's probably about a month or 2 old. It's about 3 feet long. And we caught this in the Gulf of Maine, hook and line. And you bring it up on the boat, and you can see we're drilling in the dorsal fin. It doesn't hurt. It's like getting ear pierced. I'm telling you, it's no big deal. So Amy's doing all the hard work right here. She's putting on the tag, clamping it on there, making sure it works great. Camera shot. She gives it to me. I get the money shot. I get to throw it overboard, let it go with that $5,000 tag on it. And so you just kind of pray and hope everything goes well. And so what I'm going to show you right now is a track that we've just sort of put together, and first time we've distributed it to such a wide audience. So just kind of sit back, relax. You can feel free to dance. You'll see why. You'll see the date kind of chime in at the bottom of where the shark was at that particular time. All right, so we tagged it in the Gulf of Maine. You can see it right here in October. In November, it goes up to Canada, kind of hangs out December, January. All of a sudden it makes a beeline, okay, down to Aruba. Where's the song? Jamaica. Ooh, I want to take ya. It's supposed to play, it's not playing. All right, and now back up to the East Coast, Long Island, New Jersey, that's where Snooki is. And now back to the Gulf of Maine. This shark basically went back to the exact spot where we put the tag on. Amazing. So now you can see why these tags are so important. They can give us a great information of where the shark has gone. This little shark went on a 7,000-mile round trip excursion and went down to depths of almost 2,000 feet across several international boundary lines. And so we love these tags, but we also love to connect it with other types of research. And one of the things that we do is connect it with reproduction and other life history traits. And back in the day, reproduction used to be assessed through sacrificing or killing of the individuals. And if you're a protected species like a white shark or endangered, that's a problem, right? So we look for other alternative ways to do this. And so a blood sample is a great way to get this information. You can do it from a small shark, you can do it from a larger shark, and you don't have to kill it. All right, it's like going to the doctor, getting some blood work done, no big deal. We also link these with other technologies, such as an ultrasound. And the nice thing about the University of New England is that we can actually house smaller species there and kind of practice on them to make sure that our techniques are sound, so that when we go out in the field to study these wild and larger species, we have everything refined to the best ability. And I'm going to show you some really cool things. This is actually another video. And this is an in utero image. You can see that's the little shark's mouth moving up and down, So cute, isn't that? We named it Bubbles, okay? And so Bubbles was actually born, all right? Right, I know. And then, that's my daughter, she's cute too. And so you take these technology and then you apply it to the field. And that's an undergraduate of mine, Carolyn Wheeler. And she's sitting on a 15-foot tiger shark. I would normally do this, but we've had some casualties, students getting eaten, so I don't do that anymore. And that's our collaborations with Neil Hammerschlag down at the University of Miami. So here is what she found, and that's the little tiger shark in there. It's so cute. You see the little mouth again. But here's what the exciting components are, is that when you link these together, we've got Carolyn on a tiger shark on the left, we've got a satellite tag track on the right. Now we know if that shark is pregnant, if she's going someplace to give birth. Maybe she's going to the mate or for gestation reasons. And so these are critical information that we need in order to properly manage and conserve these charismatic species. So why do I love what I do? Well, besides working with all these wonderful people here on the panel and all around the United States, it's my students. And they're in the back. Give me a wave. All right, embarrass them a little bit. My graduate and undergraduate students at the University of New England who make this work happen. You know, they're intimately, they're involved. They're part of my success. And then finally, my three little angels who have me wrapped around their fingers, and I know there's a marine biologist in there. I'm telling you. So, thank you very much for having me.
Thank you, James. Our next speaker, honored and privileged to introduce Tobey Curtis.
I've been coming to this aquarium since I was about 5 years old, so this is really pretty awesome for me to be part of this. And I definitely want to thank John and Cynthia and everybody else, Gina, for helping organize this, and everyone here is a mentor and a friend. So it's kind of cool to be part of this group. So I grew up about 50 miles from here on a little bass pond, and I grew up spending pretty much all my summers largemouth bass fishing. I'm sure maybe a lot of you do that too. And it really got me interested in fish in general. And then eventually I found out that fishing for white sharks is basically the same thing as fishing topwater for largemouth bass. And this is a photo I was fortunate enough to get in South Africa several years ago. But it's basically like pulling a jitterbug for bass. You pull these seals, you know, this is actually a live seal, or was. But it's basically the same kind of ambush predatory tactic, and it fascinated me. And so, you know, that was sort of my goal from when I was a kid, is to be, you know, fascinated with sharks, and I was kind of moving in that direction. And so when I was in college, I had the opportunity to do an internship out in California. This is a photo taken off of Point Reyes, California, and I worked with Dr. Peter Klimley out there. And I spent 2 years in his lab tagging and tracking white sharks, bat rays, some other species, and got a lot of great field experience with white sharks, which is still very memorable and awesome. And I really got to perfect my topwater techniques for white sharks. Subsequent to that, I kind of came back to the East Coast, and I got to spend some time working with juvenile sharks with the National Marine Fisheries Service Narragansett Lab. They have a long-running survey in Delaware Bay on juvenile sandbar sharks, and I got to learn how to longline and tag handle these little sharks in their nursery areas, which are, you know, estuaries along the coast. I also got the opportunity to work, like a lot of us here have kind of paid our dues, working on recreational shark fishing tournaments. You get to see some of these beautiful big fish come in, and we do what we can to collect scientific samples, and had some great experiences there. Got my hands on a lot of sharks. So from that experience, I was able to, I was hired to be a fishery observer down in Florida. And this is a photo that I took on a directed longline fishing trip down off Florida. Where they, you know, they target large sharks, and it was really eye-opening. I got to see a lot of sharks, and unfortunately see a lot of sharks killed for market. But it really got me interested in fisheries, and really got me directed me to focus my research on doing what I can to conserve and sustain these shark populations. So my time in Florida led me to my master's work at the University of Florida, where I did a tagging, tracking study on juvenile bull sharks. That's a little bull shark there. This is in the Indian River Lagoon on the east coast of Florida, if anybody knows the area. Lots of bull sharks in there. And most of my research, kind of from that point on, has focused on tagging and tracking, and it really can be boiled down to putting dots on maps. That's really what I've gotten good at, and that's one of my favorite things in shark science. You get to handle the sharks, which is fun, but then we get the data back, you put dots on maps, and that's really kind of cool. So so I've been building up my experience handling sharks, tagging sharks, putting dots on maps. And the dots on maps were enough to impress Dr. Greg Skomal, who's here. And for the last 4 or 5 years, I've been working with Greg. He was gracious enough to take me into his lab to work on his ongoing basking shark research. And as John mentioned, basking sharks are the second largest fish in the world. They're really the most common large shark in New England. And like other sharks, they've been fished across the North Atlantic and other places, and some of their populations had started to decline. There's still some occasional fishing for basking sharks in different areas, and you can see a large fin there in the on the bottom right, and some of those fins in Asian shark fin markets can sell for thousands of dollars, a single fin. So very valuable. There's a high conservation need for basking sharks, and so we wanted to take the tagging and dots on a map approach to doing what we could with basking sharks. So you may see a few more of these shots, but again, here's the satellite tag that James just talked about. And Captain Bill Shapralis from the Cape Cod Shark Hunters about to harpoon the tag into a big basking shark off of Cape Cod. This is really the fun part of the work. We get to go out and see these fish up close and see how they behave, and it's a lot of fun. But then the real work comes when you get the tag data back, and we use a couple different kinds of tags. Here's more dots on a map. These are satellite tag positions from 10 different sharks that were tracked during the summer. And each color represents a different fish. And you can really start to see just kind of even eyeballing patterns of where these sharks like to hang out. They hang out in Cape Cod Bay. They hang out in the Great South Channel. One shark really liked Vineyard Sound for some reason. But basically, these fish are they're kind of aggregating in spots where there's a lot of zooplankton. And like John said, they're really only dangerous to zooplankton, and these are plankton hotspots near Massachusetts. So they spend most of their summer and fall in these areas. And but then the big question for a lot of us was, well, where do they go in the fall, in the winter, when they leave the area? And no one had really seen many basking sharks during the winter, so it was a big question where they went. So Greg, for a number of years now, has been putting out the satellite tags, and I'm getting to work with some of this data now. And this basically summarizes the breadth of where these sharks have gone. Take that, little poor beagle. The basking sharks go all the way to South America and back. So far, I don't know, the white sharks might be giving the basking sharks a run for their money now as far as their wanderings, but these basking sharks are making really long distance migrations to the Caribbean and as far as South America and back. Straight line distance here of 4,000 miles. So the big part of my dissertation research now in Greg's lab is trying to map out where they are, but then trying to figure out why they go there. Why on earth are these sharks swimming 4,000 miles or more every year? There's got to be something really yummy down there to make the trip worthwhile. So doing what I can to try to sort that out, and that's where my career is headed in the foreseeable future. So that's all I have for tonight. Thank you.
It's an honor now to introduce Dr. Jeff Kneebone, who's going to continue some of the talk on sand tiger sharks that John mentioned.
All right, well, thank you for the great introduction, John, and thank you all for having me here. So when John initially asked me to come speak tonight, he said, okay, you got to talk about why you became a shark biologist. And for me, I thought about it for a second, and I knew right away that my answer was quite simply, fish control my brain. In fact, they have for most of my life, from being excited to get shark books for Christmas when I was a little kid, to having shark birthday cakes when I turned 30, to fueling my lifelong obsession with fishing. It was safe to say that fish are on my brain. So obviously, what's the best profession for someone who thinks about fish and sharks all the time? Well, shark biologist fits pretty well. So thankfully, I did actually get to become a shark biologist, and during my time as one, I've gotten to work with a lot of great people, many of whom are here. I've gotten to work with a lot of great species and try to answer a lot of important questions. But one of the questions and research topics that I'd like to talk to you about tonight is my work that I've done with John Chisholm and Greg Skomal of the Massachusetts Shark Research Program, looking at this species, the sand tiger. Now, John gave you a pretty good intro to the species. Just like to add a few more tidbits. Sand tigers are found in Massachusetts, but they're also found elsewhere around the world, with South Africa, Eastern and Western Australia, and Argentina and Brazil being hot spots. The other thing that John mentioned that's important about this species is that they're very commonly displayed in aquariums around the world. In fact, they used to be a staple right here at the giant ocean tank at the New England Aquarium. So many of you, if you've been here in the past, have probably actually seen these fish in person. Now, one other thing that's important to note about sand tigers, and this is an unfortunate consequence of their life history strategy, is that they're generally very susceptible to population exploitation through things like overfishing. And as a result of this scenario, the populations of the shark have been depleted around the world. And one of the areas that they've been depleted is right here off the east coast of the United States. So basically, what happened back in the late 90s is fisheries managers in the United States realized that this population was in decline and enacted some conservation measures goaled at stopping the population decline and kind of rebuilding the population. And I'm happy to say that after nearly 15 years or more of conservation efforts, that this species population is actually resurging in Massachusetts waters. And one of the places that it's rebounding is this area that some of you may recognize, and I'll refer to as Plymouth, Kingston, and Duxbury Bay. This bay is a tidal estuary that's located about 25 miles southeast of here on the western shore of Cape Cod. And what's interesting about this embayment is that about 20 or 30 years ago, when the sand tiger population was at its peak off the east coast, the sharks were never really found in this area in abundance. But starting in about 2005, we started to get a lot of reports from fishermen that they were encountering these sharks in this bay. So this was initially really surprising and then became very interesting to us. So to try to understand what these sharks may be doing here, we initiated a tagging research study back in 2008 to monitor these sharks and answer basic questions about them. So how many sharks are in here? What sizes and ages of sharks are in the bay? How long are they there? Where do they go when they leave the bay? So to do this, we employed a tagging technology called a passive acoustic telemetry, and what we found was quite remarkable. The first thing that we found was that these sharks are seasonal inhabitants of this estuary. They're basically here during the warm summer months from June to October. And in the fall, when the water temperature drops, the sharks get out of Dodge and head where? South, of course, to warmer waters. So over the wintering period, they're actually found south along the U.S. East Coast from Cape Hatteras south to central Florida. And the interesting thing is after these sharks spend the winters down south, they turn around and come right back north to New England. Now, one of the really interesting things that we found is 10 of the sharks that we tagged in this bay actually came back for up to 2 years after tagging. So that was really interesting to us because it showed us that individual sharks are seeking out this bay on an annual basis. The other thing that we found that was quite interesting, and John alluded to this before in his talk, was that all the sharks that we were tagging in this embayment were small juveniles of about age 0 to up to age 2. And this was really important to us because it showed us that, okay, these sharks are really small, so this bay may serve as a nursery area for these sharks. So for sharks, nursery areas are really important because they are areas that foster growth and survival. So you can think about how important a nursery area may be to a population that's in decline and is trying to recover. Another thing that we tried to learn from this research is what are the effects of catch and release. So again, if you remember back to John's talk, he told you that sand tigers are a prohibited species, meaning that if you catch one on rod and reel while you're fishing for any other species, you have to release the fish. So this seems like a good conservation strategy, but it really is contingent on whether or not the shark can survive the effects and the stressors associated with capture and handling. So to assess this, we took a small blood sample from both wild and captive sand tigers and tried to assess the physiological stress response of these sharks to capture and handling. And if you can see on this graph on the right, the take-home message is that while sharks are stressed by capture and handling, the important thing is that within 6 to 12 hours, they can recover from that stress and go back to doing what they do best, eating, swimming, and making little sharks. So just to wrap up, personally my favorite part about being a shark biologist is really what I just told you, all the research. I've been fortunate enough to work with a lot of great people and establish a lot of great friendships doing that research. But through that research, we can learn a lot of new things about the sharks. Through that increased knowledge, we will have better management. Through better management, we have healthier fish populations, and for me, the best part, healthier fish populations equals better fishing. So for an avid fisherman, you can't really ask for much more. So for that, thank you very much, and hope to answer any questions you have in the Q&A.
Now I'd like to introduce Dr. Lisa Natanzan. We'll be shifting over to learning a little bit about Asian growth in sharks.
Thank you, John. I work for the National Marine Fisheries Service, and I'm lucky enough to exclusively work on sharks. As to why and how I got here, I actually, unlike most of my colleagues, grew up on the West Coast, and I was just south of San Francisco, so I had the Pacific Ocean on one side and San Francisco Bay on the other. As you probably know, there's a lot of shark activity, particularly white sharks on the Farallon, so I grew up hearing about that, visiting Steinhardt Aquarium, as well as going to tide pools, and I loved sharks. And when I was about 7 years old, my mother taught me the word ichthyologist, and she said, well, you love sharks, why don't you, sharks and fish in general, why don't you study fish? And I decided, well, that's what I was going to be. Well, of course, I was very young at the time, so I digressed a little, and at one point I thought, well, maybe I'll become a doctor. My dad was a doctor, he wanted me to join his practice. But unfortunately, I liked fish better than people, so I decided it wasn't a good career choice. So then I moved on to veterinarian. I could be in medicine, but I loved dogs, so that was perfect. But there were cats. I was very allergic to them, so I didn't really like them much either. So at the time I was growing up, being an astronaut was a big thing, but they had already been to the moon. It was kind of old news, and I didn't figure they were sending me to Mars, so I said, no, wouldn't be very fun. So back then I was an avid baseball fan, Giants, not Red Sox, and I thought, well, I'll be a baseball player. But no, I probably wouldn't even make that. So I thought, well, I'll put the things together that I like the most, which is sharks and fish in general, and become a shark biologist. And by doing that, I can fish, I can play with fish, and I can go out on boats. So what could be better than that? So now how do you get there? How do you become a shark biologist once you decide that? Well, it's a combination of luck and directing your life. By the time I was 13, I was solidly settled on becoming a shark biologist, and everything I did after that went towards that. So every high school project, every college project, everything I did. I went to a college that had a boat that was run by students because I wanted to see if I could handle it on a boat and if I still enjoyed it. And when I went for my master's at Moss Landing Marine Laboratories, that's when the luck kicked in. And I was chosen to be on a grant studying the age and growth of angel sharks. So you have to volunteer, you have to be in the right place at the right time, and you have to really want it. So they asked us to speak about either a sole research project or a project of significance that we worked on. And this slide just goes to show you that we don't do anything alone. How many of us does it take to hold down this little tiger shark while it's tagged and measured? Okay? And a lot of the speakers tonight are in this picture. And a lot of the speakers have been on cruises together, shark tournaments have been mentioned, and just a shark that washes up on the beach, we call each other up and we all go. These are our go-to people if there's a shark. And that also includes some people that are in the audience today that are not speaking. That being said, my most significant research happened before I met any of these people. And that was when I started my master's. And this is an angel shark. It doesn't look like the white sharks and the exciting things everybody else is talking about, but it is a true shark. And it was a commercially exploited species off California at the time I was doing my research. And I don't know how many of you know about how to age a shark, but what we do is we take a piece of the backbone. This pointer doesn't work very well, but we take a piece of the backbone, we take one centrum out, which you can see down to the right here, and we cut it, and you can see where we have the numbers. I guess I can do the arrow here. The numbers here, we count the alternating light and dark bands, opaque and translucent bands, and one set of these, one opaque and one translucent, is considered a band pair. So back in the old days when I was doing my work, everybody assumed that these band pairs were annual. And therefore, it would be like counting rings on a tree. All right, you count the bands, you get the age of the shark, okay? So if you look at this lovely little vertebra here, and I'm sure all of you are trying to count these bands really fast before I flip the slide, but that's an angel shark vertebra, and it's beautiful. And unfortunately, it's beautiful on the outside and perfectly rotten on the inside, because after counting hundreds of these things, this is what you're supposed to get. A growth curve like this, where you have length over here and age over here, and you can determine what age it is at what size. Well, when we did the angel shark, in fact, this is what we get. And basically, it's a straight line up to maturity here, and at that point, it just goes crazy, okay? With a lot of experimentation, live grow-outs, injecting with a dye, we found out, in fact, that angel shark band pairs were deposited based on growth and not age at all. So you could have 7 of them in 1 year if the fish grew that fast, okay? Which kind of was one of the first studies that threw out the window the annual band pair deposition. That kind of directed my career and life. And now whenever I start to age a fish, which I've done since this time, it's been kind of my thing, I always question the annual band deposition, and a lot of other researchers have started doing that as well. And we have found that in a lot of other species, including the basking shark and some Australian wobegongs, band pair deposition is related to growth and not age. And in a lot of other species, including the sandbar, dusky, and sand tiger, to name a few, it's related somewhat to age up to a certain point. Usually that point's maturity, and then whatever you count on the vertebra, it's actually older. Okay, so before you call your congressman and say, why is the government funding this research to see how old a shark is, let me explain to you why it's important. Demography, which is the study of population, is important to conservation and to sustainability with fishing, okay? And demography basically is determining population, parts of the population, how old people in the population are, how many people are in the population. Well, what I do is I determine how old they are at maturity and how long they live. So from that, you can get production. How many pups do they have over the course of their mature lifespan? And that tells you whether they can replace the population or not. And that is the basis of a lot of management plans. So without this kind of research, you can't sustain fisheries and know how much you can take out or how much you have to leave in. Okay? So my favorite part of being a shark researcher is kind of everybody's favorite part. We get to fish, we get to do different things every day, we get to sometimes cut fish up, which is interesting, and we get to go out tagging and hang out with all these people. So thank you very much.
Thank you very much, Lisa. It's now my great honor to welcome Heather Marshall, who's going to be talking a little bit about stress physiology and survival, which is a topic near and dear to my heart as well.
Okay. Good evening, everyone. Thank you for being here. It's so fun to be here tonight to talk about sharks with my mentors and friends and with all of you guys. So how I got started studying sharks, this is a picture of me when I'm 3 years old with my mom up close and personal with a white shark that was brought in Montauk by the fisherman Frank Mundus. And you know, ever since that point, I guess you could say that sharks were on the brain. However, for me, I did not grow up on the water. I didn't grow up fishing, unlike many of my colleagues. from New Jersey. And so I kind of developed this irrational fear of sharks, which I had for most of my life up until my college years. And so I would read books about shark attack statistics and shark attacks and how to avoid shark attacks. And one of these books led me to this book, Close to Shore, by an author who was really good about not just talking about the shark attacks, but talking about the biology of sharks. And as I read about the biology of sharks, I was incredibly fascinated by the adaptations they had for living in an aquatic environment. And, you know, I'd be reading, I'd be like, is that true? And then I'd go online and do more research. So this led me to study or to read more books by actual shark biologists, such as this book by a very inspiring woman, Dr. Eugenie Clark, who founded the Mote Marine Laboratory in Florida. And so I didn't even know that this was a career, you know, possibility. And it was really cool learning about the shark research that could be done. And so I started thinking about that. You know, it sounded like it'd be really fun. And knowing about the conservation efforts that were perhaps needed for certain species of sharks, I decided to try it. So my first summer was working with Dr. Greg Skomal, who you'll hear from shortly, on Martha's Vineyard. My first exposure to being on boats, handling fish, fishing. It was pretty scary, but really awesome as well, and I just grew to love it. From there, I went to Mote Marine Laboratory as an intern. I got to know different other species of sharks, and that's where I really developed an interest in shark ecophysiology, understanding how the body of the shark is, how it functions and how it relates to habitat use and selection. I then began my master's with Dr. Diego Bernal, who you'll hear from next, working on shark heart biochemistry and physiology. And so here's just a picture of me harvesting a shark heart on a research cruise, which I would then analyze later in the lab. And so when I wrapped up my work with Diego, I then went on to stay with him and work on my Ph.D., which is what I'll talk about tonight, talking about the longline impacts on sandbar and dusky sharks. So sandbar and dusky sharks are two closely related species that have a history of being heavily exploited by longline commercial fisheries. So for those of you who don't know, a longline is a type of fishery gear where you basically have a long main line with many hooks attached to it, and the water is put into the gear and it's left to soak for a certain amount of time. And so because these fish were considered overfished, the fishery for them was shut down with the exception of a research fishery for the sandbar shark. However, the dusky shark and the sandbar sharks are still caught as incidental catch in longline fisheries. And so a common management strategy is that these sharks are, it's determined that they needed to be released. And so the idea is that they're going back to the population to contribute to overall population growth. However, as Jeff spoke about earlier, we don't really know what the fate of these sharks are. We don't know how they're responding to the stress and how they're handling the capture and release event. And so we wanted to try to assess the post-release mortality of these sharks. And so here's an example of how we can use these same PSAT tags, which I've circled up here, you know, which we've been learning about all these movement information about the sharks. Now we can use them to actually understand how the sharks are surviving after we release them. And I'll explain how we look at that in a moment. So we conducted a longline survey, and here's just a picture of Diego, my advisor, and my lab partner putting out and prepping our hooks to go out on the main line. Now, we did have a slight gear modification in that on our hooks we used a hook timer. And basically how these works is when a shark takes the bait, it'll start wriggling a little bit, and it'll pop the timer out, and so this starts recording. And so when we bring up the sharks, we know how long each individual shark was on the line for. So we'd bring up our sharks, put on the PSAT tags, and release them, and see what we could see. So one main goal of this project was to try to identify time thresholds at which we start seeing higher incidences of mortality from these sharks. And so we knew from previous work, a lot of work that Lisa's done on her longline cruises, that there's this threshold time of about 3 hours where you start seeing a higher incidence of mortality, specifically with the dusky shark. So just to share that bit of data with you, for sharks that were caught on, oh, I'm sorry, for 1 second, explain this figure right here. So this is how we determine if the sharks died. So basically when sharks die, they sink and they sit on the bottom. And so this is a classic sort of death profile where the shark is released, it goes down to the bottom, it sits, and then after a certain amount of time, the tag will pop off the shark and start transmitting the data. So, so just some preliminary data here. For sandbar sharks that are caught on the line for greater than 3 hours, we found 18% of at-vessel mortality plus moribund level. So moribund is a fish that is close to death. those individuals that were released, we saw 29% post-release mortality, with a combined 47% overall mortality for the sandbar shark when they were caught on the line for greater than 3 hours. For the dusky shark, the situation is a little bit more dire. The at-vessel mortality and moribund rate was 53%. And I'm sorry, at vessels when the shark is brought to the boat initially before we tag and release it. The post-release mortality is 42%, with an overwhelming 95% overall mortality for dusky sharks when they're caught on the line for greater than 3 hours. So obviously this is a situation that now we can share this information with fisheries managers to try to perhaps remedy the situation and, you know, help create and sustain a fishery. Okay, so we have this hook time information, which is basically from the hook timers, but how does that carry over into management? You know, you're going to manage by possibly augmenting soak time. So this is just a graph showing the correlation of hook time that the sharks were on the line and the soak time, which is the amount of time that each set was in the water for. So the straight line there shows the correlation, and we were really happy to find a really nice strong correlation indicating that our hook time information was representative of the soak time. And so we can share this information, and for example, if we wanted to target that 3-hour limit, if we were to reduce soak times to 3 hours on the Longline, we could perhaps have a 46% mortality reduction in sandbars and a 60% mortality reduction in duskies. So this is just an example of how we can use these tags and this technology to help with fisheries management. And we consider this project to be rather timely, as the dusky shark is currently being considered for the Endangered Species Act. Okay, and as to why I love this job, I would say that on top of just learning about sharks and the science itself, every day is different. It's a very dynamic job. Obviously, the field work is a lot of fun. It's beautiful. You get to travel to amazing places. Working with sharks is really incredible. It's really humbling and awe-inspiring to put your hands on a shark and be able to work with them. It's great to be able to do lab work as well, have visiting scientists come in, do work together, talk about possible projects. And I'd like to just also take a moment to talk about the Gills Club, which I'm a co-founder of along with Cynthia here and a couple of other women. I'm so happy to see so many young girls in the audience tonight, and it's really been a wonderful program to have helped start it and be a part of. We have events where you know, we can give girls hands-on activities where they get to know shark research. And so here are just a couple more pictures of the event where we taught the girls about shark tagging. And so it's really been a wonderful experience being able to do that. So if there are any women or girls out there who are interested in sharks, please check out our website. All right, with that, that's all I have. Thank you.
So now I would like to introduce Dr. Diego Bernal, who's our next speaker, our second to last speaker of the event.
They just told me I have 2 minutes, but Dr. Skomal's after me, so I'm taking all his time too. How did I get started with this? I want to know how sharks fly. To me, this is really telling of why I do what I do. It's really looking at animals that are really extreme, that do things they're not supposed to do. This is a Thresher shark that's jumping about 15 feet off the water. And that just shows you the power these animals have to swim really, really quickly and jump out of the water. And that drew my attention since I was a small kid. I grew up in the city, and it wasn't until I got to grad school that I started working with sharks. So, you know, there's time for everybody to start working with these animals. But this is what we enjoy. It comes over and over again. Yeah, I mean, we love fishing. If you want to be on the water, this is the way to do it. It's how we kind of all get hooked on this field. But it's not really nice. This is a trip we did down to Mexico one time. Everybody was throwing up over the side, and you can't come home, and the storm got worse, and hopefully you don't get dizzy. But this is what we do, and it's a lot of fun even to be out there in those conditions when you get home, and it's a good memory. What do I do, and what does my lab do? We're shark physiologists, which is really a fancy term for a mechanic. We study cars, and unlike Click and Clack from NPR, we don't do gremlins. We do something a little bit more exciting. We study those. We pick and choose our species to be the really extreme, fast-moving fish that can do really good things, so with hot rods. And we study hot rods in many different aspects of them. So what I'm going to show you next is a picture of a mako shark, because they're also really powerful. They jump out of the water, they follow their prey, they eat tuna. They eat other fast-moving sharks, get out of their way, and that's kind of what we do. But when we look at a shark in the lab, what we really interests us is what do we study? And the analogy is they have a computer, they have these air intake carburetors, they have the engines, and they have the thrust machines. The computer's overrated. Nobody needs a brain. So we really just study the machine and the thrust propulsor and then sort of the intake and everything else in there. And I'll be talking about that thrust, that tail for today. This is a normal shark, even though I just always an extreme turbo shark. It's a normal shark. It's pretty boring. It has a big tail, big body. It's all about swimming for them. And the main trick for them is that when they move forward, the tail pushes them down. It's a thing we can talk about over a couple of beers later on. The body pushes them up, but they go forward all the time. And depending on the size of the tail, the bigger tail you have, if it's really long on the top, it pushes you down even more. Which leads me to today's talk, which is the Thresher sharks. They're just weird. They have this huge tail that's as long as the body, which is remarkable. If you deal with an animal that's 8 feet long, there's another 8 feet of tail behind it. It's huge, and that pushes them down a lot. So if you look at the arrows really quickly, they're huge arrows of pushing them down. The fins in the body bring them up like an airplane, but they still go forward. And so I know it's a cartoon, but this does them justice. Look at the tail. That doesn't even fit in the IMAX screen. John, you told me it was going to be a big screen. It's huge. Look at the pectoral fins, the big side wings. They're absolutely massive. And so I'll be talking about Thresher sharks because the tail is what a remarkable feature of this group. There are 3 species of Thresher sharks, live in very different parts of the world, different depths, different temperatures, all have this huge tail. All fishermen kind of knew what tail was used for, but nobody actually ever saw them use it, and we thought it was for feeding. Well, obviously, the first thing you see a tail like that, it has to be used for locomotion. So there they are. These are some threshers we caught in California. Wrong coast, I'm sorry. But you can see them swimming at the surface after we've tagged them, so they're kind of groggy, and we get some great image. The tail is being used for locomotion, and that's great. But what was really cool is that we had a lot of time on our hands. So we're able to go down and start looking for Thresher sharks underwater. And so we started filming for Thresher sharks going past, this is a school of anchovy. This is their favorite bait. And we're put a camera by our fishing gear, and you'll see 2 little white dots somewhere in there. That's our fishing gear. And we sit there and we look and we look, and you get bored and you look, and every now and then, there they come. And what I want you to focus on is that tail. That tail is really kind of acting almost like a whip. And so our objective from this study was to figure out if they're using their tail for swimming or for swimming and for hunting. And it turns out that the tail is a feeding appendage. They use it to, and they're really smart, they whip their bait and they hit the bait with the tail, they stun it, and they come back and they eat it. Their teeth are really, really small. They're kind of not shark-like, they're really wimpy. But the tail is the really, really aggressive tool. So we're able to go in there and look at the tail of these Thresher sharks and do all sorts of measurements on video and pictures and figure out that in less than a half a second, they can come up to their prey, they can swing it, they can hit the bait, and sometimes they hit it so hard they split in half. And they just come back and they eat it. So they're kind of saving energy. What's remarkable, I didn't show you the 2 hours of footage that we have on these, is that we're going really slow. Most often they came up to the bait, saw it, and said no, and they just left. So it's a rare occurrence where they do this, but the tail is used for feeding, which is good, and it's really bad. Because this is what we usually see in sharks. When you go out there and you hook a shark when you're fishing, you hook him by the mouth, which is good because you can bring him to the boat, you can take the hook out, and you can let the fish go. But in the Thresher sharks, because they're using their tail as a feeding appendage, that's what you get. You catch them by the tail a lot. And so this is a problem that we've been trying to solve is first of all, what happens to the sharks when you catch them by the tail? How long can you fight them if you're going to be dragging them backwards and they can't breathe? And so we've been trying to work with that scenario, and we're getting some really interesting data that's not for this conversation, but really I've gone from the swimming and the animal to its feeding, and then coming back to how they use the swimming for feeding and how the consequence of that when they interact with humans in the fishery. I want to leave you with this one beautiful image of a Thresher shark swimming away from the boat with a $5,000 tag that we never saw again. And I think the most favorite part is dealing with these animals. The honest truth, the most favorite part of what we do is dealing with students and with colleagues. This is really important part of our sanity, is getting out in the water. And every now and then, the favorite part is this. Anybody know where that is? That's Greenland. We were fishing for sharks in Greenland last year, and this is where our work takes us, all the way all over the world. Thank you.
Last but not least, it's great to have Dr. Greg Skomal here. I don't think any of you know him.
Is this working? Yes. Okay. Well, thank you. They said they picked this order based on height. Yeah, I believe it. What great stuff. It's hard to follow all this up, but I'm lucky. I get to talk about white sharks, right? And we won't go to that just yet. So today I'm going to give you, I have to give you a really short story. And anyone who's ever seen me before knows that my stories tend to be long. And I go on and on and on. So I've had to condense this into what amounts to probably less than 30 seconds. But it's going to go for at least 6 minutes, so don't wave any flags. And because no one's after me, I can just keep going and going and going. But so today I'm going to talk about white sharks, and I'm going to talk about technology, and I'll talk a little bit about me. And give you a sense of what's changing, what's changed dramatically over the course of the last well, for me, 30 plus years, and particularly with regard to white sharks over the last 5 years. That's me in 1975. I was graduating from 8th grade. I don't even know if they call it what do they call it, middle school now or something. And I didn't have a clue. I was a kid who grew up on Long Island Sound, which was not the most pristine body of water. But I also grew up in front of a television watching Jacques Cousteau, and I was obsessed with Jacques Cousteau. And I knew at that age that I wanted to not only become a marine biologist, having not spent a lot of time in the water, but truly loving what I was seeing Mr. Cousteau come out with, but also specifically I wanted to study sharks. And so over the course of the last 39 years, I morphed into this guy, which is considerably older, but holding a shark. And now I've had the opportunity to not only work with these folks, which as you can see, is the best of the best, but also to work with those animals right there. Many, many different species doing many kinds of studies that are very similar, and collaborating with these folks on physiology and ecology and natural history. What's also changed over the course of those 39 years are the tools we've got to use, which has been really, really fascinating. You know, early on, Lisa talked about the age and growth work that she does. A lot of researchers continue to do it. I've done it. We're working on a paper right now. She's mad at me because I haven't finished it with her. But all these guys are mad at me at some point because I'm not finishing things. And a lot of our samples were coming from dead fish. You've seen a lot of dead fish tonight because we need to sample dead fish to learn about various aspects of their natural history. So in the case of white sharks, over the course of the last, several decades, we've seen little white sharks, and we've seen really big white sharks. And we've taken samples from them, but it takes a long time to get these samples in the case of white sharks because they're extremely elusive animals in this part of the world. We're in the western North Atlantic, we're in the North Atlantic, where white sharks are very, very difficult to find, or were very difficult to find. That's me 4 years later in 1979, graduating from high school. I still had no clue. I just didn't realize it. I took a few advanced biology courses, and I was about to embark on a college career at the University of Rhode Island. And I was ambitious. And as Lisa said, this kind of path requires ambition, drive, tolerance, maybe a little bit of your own money in some cases, volunteering, all those things that get successful people to where they need to be. Also in 1979, coincidentally, a scientist at the Woods Hole Oceanographic Institution named Frank Carey, working with some of my mentors, and he was one of my mentors at the time, tracked for the very first time a white shark, putting an acoustic tag on a white shark. Now, we've heard a lot about lots of different technologies tonight. The very first one to really be used in terms of electronic tags was his acoustic tag, and Frank built his own tags. And he had this rare opportunity where he found a white shark feeding on a dead whale, and he put this tag in it, and for 3.5 days, they followed it. getting for the very first time insights into the behavior of this animal. Because there's one thing about studying dead fish you don't get, and that's basically the ecology and the behavior of that animal. And that's where we need to study live animals. And Frank was the first to do this on a white shark, and it happened to be right off of Long Island, where he tracked this animal down the coast of the island for these 3.5 days. I'm going to blow that image up because I want to put it in perspective to what we've got now with the changing technology. So this is just a glimpse, a 3.5-day glimpse into the biology of this animal. And of course, the years that followed, as technology changed, as tools became available to us as scientists, places where you could find white sharks like the Farallon Islands, South Australia, South Africa, Guadalupe Island. Scientists were using these tools to study white sharks and learning amazing things. Well, we've been pretty lucky here in New England to suddenly have the occurrence, and I don't mean suddenly in the sense of overnight, but over the course of the last decade, have the occurrence of white sharks occur very, very consistently off of Cape Cod. And now we have been able to, over the last 5 years, tag 39 of these individuals using a variety of technologies. You see the acoustic tags listed there. It's the same technology Frank used. Jeff talked about it. It's been adapted to passive acoustic telemetry, very same principles. James, Heather, they've talked about the pop-up satellite tags we're using. We've put a bunch of those out on white sharks, real-time satellite tags, collaborating with our friends on the OSearch, where we could track the sharks in near real time. And I believe that James was doing that with the tiger sharks down in the Bahamas. And then I'm going to finish this talk with the robot tag. Technology continues to change so rapidly, and I often say it. You know, when you see that picture of me in 1979 going into college, we didn't have laptop computers. But now we're putting them on fish. And that's really, really amazing to me. It's really rewarding. It's really exciting, as you could probably tell. So this is what we've learned about white sharks over the last 5 years. And look at the breadth of these movements, the dynamics of these movements. You saw the poor beagle, you saw the basking shark. The more sharks we tag with this technology, the more that we realize we don't know anything about them. They go to places we never thought they went. Basking sharks to the Bahamas, white sharks to the mid-Atlantic ridge, diving to depths as great as 3,000 feet every day. We didn't know this because we didn't have the tools. And I've been able to live through this period of dramatic technological growth. And for me, taking it to the next level is always very important and very exciting. So the next level for us was to try to use some kind of tool that would tell us what these sharks are doing when they're in these remote areas. Now, many of you have heard about drones. It's the catchphrase these days. Certainly when the airliner went down a few weeks ago, there's a lot of discussion about sending a drone down to find it. This drone technology has been evolving over the last 2 decades. I got together with my colleagues at the Woods Hole Oceanographic Institution with some funding from the Discovery Channel to try to adapt a drone to follow a fish. It had never been done before, and indeed it wasn't going to be easy. As a matter of fact, I didn't even think it would work, quite frankly. But we didn't tell the folks at Discovery that, of course. You could see here pictured the Remus 100 that Woods Hole Oceanographic builds and uses. It's a smaller drone. It goes to about 100 meters, and it's perfect for our uses because we could carry it, lift it around, take it on small boats, and have it chase sharks. But in order for the drone to follow the shark, you got to put something on the shark. You can't just program the robot to identify a fish. It doesn't work that way. And we got really, really good at putting things on sharks. All these people have gotten really good at that. And so we built this transponder. And the transponder communicates two-way communication between the robot itself and the transponder. And basically think of it as a very sophisticated game of Marco Polo. The AUV, the robot says Marco, the transponder says Polo, and the AUV follows it. So conceivably, we thought it might work. And indeed, in 2012, we were able to track 4 white sharks off the coast of Cape Cod very, very successfully, following them, not only following them, but actually filming them and their behavior. And this aired on Discovery Channel last year. This year, Discovery was so excited about what we achieved, as was Woods Hole, that they actually decided to take the robot to a very cool place where white sharks occur in large numbers. And that is Guadalupe Island off the coast of Mexico, where we had very, very clear water up to 100 meters of visibility, lots and lots of white sharks to follow them. As far as what drives me and what I do, it's producing information, working with these collaborators to produce information that goes directly into conservation and management of shark populations at sustainable levels. And being able to come out here and actually share it to the public and bring you into the fold is equally as rewarding. So thanks a lot for coming out tonight. I hope to meet with you later.